Okay, so we talked a little bit earlier about polyploidy, and this is going to be where there are more than two sets of the chromosomes. So in humans, we're going to be a diploid. We get one set of the chromosomes from our mother, one from our father. In some cases, the organism can have multiple copies. And so polyploidy is going to be pretty infrequent in many animals. Some exceptions might be lizards, amphibians, and fish. But it's actually fairly common in plant species. Now the only catch is that the odd number of chromosome sets is not going to be reliably maintained generation to generation, because if you did that, you would not be able to produce genetically balanced gametes. But if you're a 2N, a 4N, a 6N, and so on, you're going to have equivalent numbers. You're going to be able to balance that a little bit better. You can distinguish between two types of polyploidy based on the genetic origin of the extra chromosome sets. You can have autopolyploidy, and what happens here is you have addition of one or more extra sets of chromosomes, which happen to be identical to the normal haploid complement. Allopolyploidy happens because you have a combination of chromosome sets from different species. Maybe they're very, very closely related, and it's basically a result of hybridization. So let's look a little bit closer because these are big words. A lot of people need to see the figures. What's actually going on here? Okay, so in autopolyploidy, if something is diploid, here's three different types of chromosomes. Chromosome pair 1, chromosome pair 2, chromosome pair 3. You may have some event where the cell goes into interphase, you duplicate the DNA, but for some reason it never undergoes mitosis, and you never get a splitting of those cells. Okay? Instead, what can happen is you have these extra copies. If it's triploid, you're going to have 3 copies of each of these chromosomes. If it's tetraploid, you've got 4 copies of each of these chromosomes. Okay? But they're all from the same original source. And that's indicated because they're all the same color here. In contrast, in the allopolyploidy, you're going to have 2 different but very closely related species typically interacting. And you can see here a diploid individual with a diploid individual, and you're going to have a tetraploid offspring. So you've got 2 copies of this particular chromosome from that parent, 2 copies from that parent, 2 copies from that parent, 2 copies from that parent. Okay. So how does it happen? Well, I mentioned before, it can arise naturally if these chromosomes replicate, but the parent cell never divides. And instead, maybe the cell is going to re-enter early interphase, going to duplicate the DNA again. It is, or may be, an advantage, particularly in plants. In some circumstances, we've found that polyploid plants tend to be bigger, they tend to grow more vigorously, and so they may have just a natural advantage if they do this. In addition, hybrids, some of the offspring are going to be sterile if those chromosomes are not compatible for synapsis. So say you've got chromosome A from one parent and chromosome B from a separate parent. If those are unable to undergo synapsis or crossing over during meiosis, that offspring is going to be infertile. And so you might see this with just different organisms that happen to be together that, for example, is it a horse and a donkey together are going to make a mule, and that mule is going to be sterile. If, however, that genetic information undergoes a doubling, so now you have 2 A's and 2 B's, that may help to circumvent this problem because the 2 A's can synapse, the 2 B's can synapse, and so on, and so there's not really as much of a problem. Hybrids are also used quite a bit in agriculture. So one relatively recent example, wheat and rye together make something called Triticale. So wheat is very high yield. Rye can grow in some pretty unfavorable environments, so you put them together in unfavorable environments, you can grow a lot of food. Okay? Another benefit, things like wheat might be low in the amino acid lysine. Rye happens to have lots of lysine. Okay? So that would improve the nutrition of the plant. And you can do this with any sort of related plant, and we have done that in agriculture, and we've made great leaps and bounds. So it's a little confusing to people. What exactly is going on here? How can it be fertile or not fertile? So let's take a little bit closer look at that. Okay, so let's say species 1, okay, it's got 2 genes, okay, it's got A here and lowercase a, it's got capital B and lowercase b. when it forms gametes for this. Okay, A of course needs to be taken down to here, capital B needs to go here. Out of these you're going to take one from the lowercase a1, a2, a3, here you're going to take b1, b2. When they come together through fertilization, you are now going to have a sterile hybrid. Okay, but you're going to have one copy of each of all these different chromosomes, or genes, if that's what we're talking about. So then, say you have now a chromosome doubling. Okay, now you have 2 copies of A1, 2 copies of A2, and so on. Now this organism might be fertile. Okay, so now you can produce gametes that are going to be genetically balanced.